all, so much to Nick for organising today. And Jane, sorry, yeah, the presentations have been amazing. So I just got back from overseas this week, giving a version of this talk to a veterinary immunology conference in Vienna. And safe to say, hardly anyone in the audience was familiar with devils, so it was quite fun introducing them. But I don't think that's necessary for this group. And Andy has given a good background to DFTD, so I'll skip this slide as well. But I will just give a bit of background to MHC that's relevant for the study that I'm going to share with you. So the major histocompatibility complex, class 1, or MHC1 molecule is present on all nucleated cells in all vertebrate species, and its primary role is to allow the immune system to distinguish between self and non-self. There are 3 MHC genes, A, B, and C. Each has 2 alleles, and it's this MHC diversity in a species which is critical for the immune fitness of a species and its ability to respond to a whole variety of pathogens. So as far as DFTD is concerned, the primary way it escapes the immune response of the devil is by downregulating those MHC molecules. But that's reversible by exposing the tumour cells to an inflammatory cytokine such as interferon gamma. And that's really easy to do in the laboratory. There's also evidence that it occurs in nature. So that's, the background. This study that I'm sharing with you today is actually, it's the culmination of a long-standing collaboration that we have had with the Australasian Wildlife Genomics Group from University of Sydney, and also the Save the Tassie Devil program. And the hypothesis that we tested, which is that devils with an MHC type that's quite different to the MHC type of the tumour cell would be those devils that are most likely to have an immune response against DFTD. Has actually been a hypothesis that's been around ever since it was realised that DFTD is a transmissible cancer, because being a transmissible cancer means it's an allograft, which means that it has a lot of similarities with human organ transplants. And, it's been known for decades that if there's a mismatch between the MHC type of an organ donor and recipient, then it's guaranteed that there'll be immune-mediated rejection of that organ. But it's only recently that we've had the methods to, as far as doing the deep sequencing of the MHC of the devils, that we've been able to thoroughly test this hypothesis. So our current study, has built on results from 3 previous studies. The first of those was this 2016 paper, where we showed that wild devils can mount an immune response against the tumour, and that immune response is in the form of serum antibodies against the tumour cells that have been treated with the interferon gamma. So I think we have 52 devils in this study, and 6 of those had those serum antibodies. And 4 of those devils had undergone tumour regression. So, when a devil gets a tumour, the vast majority of those devils die within 6 to 12 months of that tumour first appearing, but there are some remarkable cases of tumour regression. What we didn't know in this study is whether the serum antibodies were recognising the MHC complex, or if they were binding to some other molecule that had been upregulated by the interferon gamma. But then in 2021, Chrissy Ong in our group did this really cool study where she knocked the MHC molecule out of the tumour cells using CRISPR, and was able to demonstrate that it was indeed the MHC complex that the antibodies are binding to. And as Andy said earlier, Chrissy's now taking a deep dive into exactly which part of that MHC complex the antibodies are recognising, and whether they will be useful targets for the vaccine. The other thing that's worth mentioning here is, once again, in human transplant medicine, when you do get immune-mediated rejection of the graft of the organ, the key mechanism for that rejection is the development of antibodies against the donor MHC. And then around the same time as Chrissy's experiment, the genomics group perfected their method for sequencing the MHC genes of the devil. So with that, we took 116 wild devils that we had both immune serum data from and MHC typing of. And then we excluded all of the healthy devils that had no antibodies, because we wanted to be sure that the devils in the final analysis had been exposed to the tumour. So we ended up with 61 wild devils, and 25 of those devils had serum antibody. 13 of the 25 were healthy, and the remaining 12 had varying stages of the tumour. So this figure just shows 5 examples of those 25 devils. The top row has the devil's identification and the year they were sampled. The next row obviously is the photos of the lesions if they were present. And then the last 2 rows are the antibody responses. So the bottom row is antibody response against the treated cells. And the row above that is antibodies against the untreated or MHC negative tumour cells. And the black peaks are the negative or naive serum controls, and the red peaks are the test serum. So any shift to the right of the black is a positive result for the antibody. So I'll just quickly go through a couple of these examples. The first one, KI, she's one of those examples of a tumour regression. And she was positive for antibodies against MHC. The devil number 3, SY, she was trapped, I think 3 times over 4 years, and each time she was healthy and each time she had antibodies against MHC. The last devil, number 5, he had quite an established tumour, and he was one of only 4 devils that had the antibodies against both MHC positive and MHC negative cells. So with that, the genomics group carried out a series of generalised linear models, and they found that there is a correlation between the antibody response in a particular MHC type at the UA genes. And that's illustrated by this figure. So all of the devil MHC alleles are grouped into 13 supertypes. And for our data set, the UA alleles were all of supertype 1, 2, 7, or they weren't present at all, so they were deleted. Now, along the bottom row, you've got all those 61 devils in the study, and the devils that had the immune response are marked by that black squares or circles above them. And then the 2 DFT1 cell lines that were MHC typed are there circled in black. And what's immediately apparent is that the majority of devils that had an immune response had a deletion of both those UA alleles. And in fact, there's 21 of the 25 devils that had an antibody response had at least 1 UA deletion. And there's just 1 devil with an antibody response that shared both those supertype 2 alleles with the tumour. So then we thought that if that immune response is beneficial for the devils, and if it is associated with the UA deletion, then there should be evidence for selection for that UA deletion across the broader devil population. And so we got another 537 devils that we had ear biopsies from that had been collected across 9 field sites across Tassie over an almost 10-year period. And they were MHC typed. And what we found in general is that there was a trend for the frequency of the UA deletion to be increasing and a trend for the frequency of the UA supertype 2 to be decreasing. And this was at most of those field sites, except for Woolnorth, which is up in the far northwest corner of Tassie, which at the time of collection was disease-free. And the frequency of the UA deletion and supertype 2 tend to be fairly steady at that negative disease site. The last bit of evidence that supports the significance of that UA deletion is when you look at the frequency of the UA alleles across the different age classes. So, devils only live to a maximum of 7 years in the wild. And prior to DFTD, it was not uncommon to find devils that were, 5 or 6 years old. But in populations where DFTD is established, it's unusual to find devils older than 3 years. But if you look at just the last 3 columns of this table, you'll see that the frequency of the supertype 2 alleles, which are shared with DFT1, is 0 at all the field sites except for the disease-negative wool north site. And then the converse applies to the frequency of the UA deletion, which is between 50 and 100% at all of those sites, except once again for wool north. So in summary, yes, there is a particular MHC type associated with the immune response against DFTD, and there is evidence that this UA deletion is being selected for. So while this might bode well for the devils in general responding to DFT1, it would result in reduced MHC diversity of the species, which of course would make the species more vulnerable to other current or emerging pathogens. And so if we can get a vaccine that protects the devils against DFTD, it'll not only help prevent disease, but also preserve MHC diversity. And that's actually it.